they junior lecturer. We call it here. We call it junior lecturer in Philippines. You can see their face. They are still fresh, yeah. And they will share the you see what is major crops in the Philippines. Major crops or plant disease crop. They will share okay. For the first, we will know what the crops in Philippines, and then uh, uh, associate or assistant. I couldn't distinguish. Assistant professor of Rejuso, Mr. Rejuso will uh, talk about common insect pests in the Philippines, and then uh, associate professor Mr. Betani or Mr. Macasero. Macasero. Mr. Macasero will talk about disease diagnosis. Of course, during talking about disease diagnosis, we will know what kind of disease in Philippines and so on. So we hope it will broaden your knowledge about our neighbor Philippines. Jadi teman-teman, hari ini kita dapat tamu dari Filipina. Mereka adalah junior lecturer, dosen-dosen muda di sana, dan saat ini sedang istilahnya benchmarking, ingin melihat bagaimana kita belajar di sini. Mereka juga ingin melihat lab kurikulum kita, laboratorium kita seperti apa, ya, sehingga mereka datang ke sini. Semoga dengan melihat face mereka itu juga menjadi inspirasi untuk Anda, ya. Saya juga ingin jadi dosen misalnya, gitu. Tidak harus di IPB, di banyak tempat. Kalau semuanya jadi dosen IPB satu kelas ini gawat juga gitu ya. Gak cukup juga ininya. Tapi kami harapkan itu menjadi motivasi untuk Anda. Kemudian tadi saya katakan kalau biasanya nanti teman-teman akan ke lapangan melihat, bertemu dengan petani, membahas permasalahan mereka. Nah, hari ini kita akan melihat bagaimana tanaman-tanaman apa saja yang ada di Filipina, kemudian hama-hama apa yang bisa ditemukan di Filipina, lalu penyakitnya juga, ya. Jangan malu-malu untuk bertanya, karena ini adalah diskusi. Ada sponsor. Siapa yang bertanya dapat ini. Jangan malu-malu untuk bertanya. Ya, someone who have a question, we will give this. Ada sponsor. Souvenir. Siapa yang bertanya dapat ini. Wow, this is from Philippines. Yeah. Okay, so someone have a question. We will give this. And we are sorry that Pak Hermano, you met him already, yeah. He is not in a good condition, so he asked me to replace him. Okay. Sorry that Pak Hermano, you met him already, yeah. Okay. First is Mr. Enabore. Are you ready? Okay. Time is yours. Okay, you can read it. Mr. Enabore is entomologist actually, and he has a BS in agriculture, in especially in entomology, specialized in insect ecology, and then yeah, he is now in as a I always said junior lecturer in the University of the Philippines, Los Baños, and now he is working in insect ecology laboratory. Ya, so he is in BS, mungkin kayak S1-nya kita ya. BS is bachelor in agriculture and specialized in entomology. Okay, time is yours.
Okay, everyone, can you please can you hear me at the back? Okay, salamat siang and good afternoon. Nama saya Rex Joseph DJ Navore. Am I saying it right? Am I saying correctly? Okay. My name is Rex. Just call me Rex. I'm an instructor from IBPUPLB, and for this afternoon, I'm going to talk about the major crops of the Philippines. Indonesia and the Philippines share the same crops. Actually, we eat the same crops, and we have the same crops as well. And actually, our climate are quite similar. That's why the pest that we have in our country are most likely the same as what you are seeing here in your country. Next slide, please. Now, just some statistics about the Philippine agriculture. Philippine agriculture actually contributes to 9.6% of the country's gross domestic product way back in 2021. And it's actually experiencing a very small decline, just small decline. 0.3% decrease in overall production. That includes our crop production, our production in agri-fisheries as well, and also our livestock production. It's just very small, 0.3%. And in terms of crop production, the entire country, our country, the Philippines, experienced 2.2% slight increase in overall production. And it employs a large portion of the country's labor force. It employs 24.2% of Filipino who are working in our country. That's why in our country, I think it's the same as yours. Agriculture is a very fundamental portion or a very vital part of our economy. It employs lots of people, feeds a lot of Filipinos, and it's a very important part of our culture and also our identity as well. Next part, please. And rice is our favorite crop. I think it's called nasi in your in Bahasa. Is it nasi? Okay, nasi. We love nasi, and in the Philippines, we love rice as well. Actually, in the Philippines, the statistics is I think it's 2016 to 2021. The entire country was able to produce 19.29 million metric tons of rice, and we have two major ways of growing rice. We have rain-fed rice and irrigated rice. So for rain-fed rice, we just grow the rice and we left it on the field. We have no irrigation. The water or the farm is usually reliant on the rain, no, for water source. But for rain-fed rice, we of course have irrigation that supplies water for our rice fields. And next slide, please. Okay. Sorry. Okay. Here's the map of the Philippines. I think Indonesia is found lower near the equator, and here's Kalimantan. And the ones that you're seeing in the map are the green ones. So the higher the intensity of the green color, it means that the higher the amount of rice production is in our country. So in the Philippines, the major rice producing areas are found in Luzon, in this portion right here, and also in the middle in Visayas, and also in some portions of Mindanao. And we have lots of terminologies for rice. Our country is our Filipinos really love rice, so we have lots of terminologies for rice. So I don't know if the only bahasa that I know for rice is nasi. I also heard of nasi puti. So in the Philippines, in Filipino, the term for rice, the actual rice plant is palay. Can you say palay? palay. We call it palay in the Philippines. Now, lots of Filipinos rely on palay for food. And it's the one that usually fills our hungry stomach when we are starving, no? And when palay or when the rice is harvested already and it has been processed, we call it bigas. That's our term. Oh, okay, okay. And when palay or when the rice is harvested, Bras. Okay, okay. So I learned some bahasa today. No, okay, thank you. And bigas is very expensive currently in our country, no? But nevertheless, we Filipinos are continuously striving to feed our families. Next slide, please. And when we have already cooked the rice, we call it sinaing. And sinaing. Oh, this is nasi. Okay, so we have we call it sinaing. Can you say sinaing? Sinaing. So actually, Indonesia or Bahasa and Filipino has the same pronunciation. No? We usually pronounce the words on the same way. But nevertheless, some Filipinos love this burnt rice during the cooking process, and we call it totong. We really love it because it's sometimes crunchy and chewy as well. Okay, next slide, please. Some Filipinos love this. Next slide. Okay, and when we have already, and sometimes the leftover rice is fried next day. We call it sinangag. I think you call it nasi goreng. Am I correct? Yes, I love nasi goreng. Anyway, and we have some rice who are called this who are cooked as desserts, and we call them kakaning. That's the umbrella term for rice meals who we eat after or as sweets after a heavy meal. And next slide, please. And the Philippines is actually the first country who have approved the cultivation of golden rice. Do you know golden rice? Golden rice is a new variety of rice plant that was a product of a research collaboration. And the golden rice was made or was bred in order to address the growing vitamin A deficiency in our country. Nowadays, a lot of young Filipinos, especially the children, are experiencing vitamin A deficiency. And this usually leads to very weak eyesight. And that's why a lot of young Filipinos nowadays are experiencing or are having difficulties when it comes to seeing or having vision, because vitamin A is very fundamental or is very vital for the complete development or for the complete functioning of our eyes. And this variety is now is now foreseen as an answer to that problem. And hopefully, a lot of Filipinos will eventually buy it in order to help solve or address the said problem. Hopefully, in Indonesia as well, you will be able to grow it. But nevertheless, in many countries around the world, the golden rice is now currently at approval or a trial phase. But in our country, it's being sold already. Next slide, please. Okay, now let's move on to corn. Corn is also another vital crop in our country. What do you call the corn in Bahasa? Jagum? Okay, so we also love corn, but majority of the corn that is produced in the Philippines is actually for animal feeds. We grow them, the yellow corn, we grow them as feeds for animals, especially for pigs and also for other livestock species as well. And we have 8.12 million metric tons of corn produced in 2021, and in the whole country, 74% are actually yellow corn, and the rest, 26%, are white corn. White corn is the one which we as Filipinos love to eat. And again, let's go back to the map of the Philippines. Majority of the corn producing areas in the country are found in the north. That's region 2. And the Philippines is actually divided into 18 regions and in 81 provinces. Okay? But always remember that for corn, the top producing areas in the Philippines are found in the north, the region 2, and also this central region here, the region 10. Okay, next slide, please. Okay, next slide. So that's white corn and that's the yellow corn. Next slide. Okay, and now let's move on to banana. It's Musa Caminata. I think you call it pisang, if I'm correct. Okay. We love banana as well. We love banana. But majority of bananas in the Philippines are exported to other countries. No? The major countries are usually Japan and parts of Europe. They love the Cavendish banana that are grown in Mindanao or the lower portion or the southern portion of the country. The main variety that we are growing is the Cavendish variety, followed by Saba and Lakatan. I think Saba is, I've seen Saba in Indonesia. Saba is a Filipino term. It's the one that the variety that we are using for cooking. Okay, yeah. Yes, the plantain. We use them for cooking. We call it Saba in the Philippines. Next slide, please. So, in the Philippines, the major producing area of banana is found in the southern portion. This region here. There's a lot of international companies there. So, Mifru, we also have Tadeco, we also have Del Monte, and they are growing lots of bananas. Not only for Filipinos, but also for other countries as well, which are exporting from us. Next slide, please. And next we have mango. Mango is one of the best crop for Filipinos because we love it. And actually, out of the entire production of mango in our country, only 6% are exported outside of the country. So the 94% are actually consumed by a lot of Filipinos. That's how Filipinos love their mangoes. And we actually produce 39.25 thousand metric tons of mango in the recent years. And majority are actually Carabao mango, the one that you are seeing, the large ones in our country. Okay, next slide. Okay, and the majority of the mango producing regions in our country is found here and also on the southern part, also in Mindanao. The Philippines is actually a typhoon pathway for a lot of typhoons that are formed in the Pacific Ocean. I think, do you also have typhoons here in Indonesia? Not many, but not many or very few, just few typhoons. Yes, very few. In our country, every month there's sure to be at least one typhoon that will enter our country, and our country is also the one who have quote-unquote welcomed the most powerful typhoon in recorded history. I think that's called Typhoon Haiyan. It caused a lot of economic loss for the entire country, not only agricultural industry, but also in a lot of our economic sectors are damaged as well because of typhoon. And the usual typhoon pathways are usually right here. Okay, so a lot of typhoons that are formed from the Pacific Ocean are likes to pass the Philippines in this path, and that's why this area right here is usually spared from typhoon. That's why a lot of annual and also perennial crops in our country are grown in this region. This island is called Mindanao, but we also have lots of island groups here. We have Soluta, Witawi, but that reason, the absence of typhoon in Mindanao, but due to climate change, we are also experiencing, I think in English, what do you call calamansi in Bahasa? Okay. Okay, we love calamansi because it's actually used for seasoning in a lot of fried food. So we Filipinos love to fry our food. We fry fish, pork, and many different animals, and we usually dip them in seasonings that contain calamansi, and that's why we also produce them in large numbers. And in the Philippines, we have produced 108.69 million metric tons of calamansi, so that's a lot. And again, we use it for cooking, for drinks as well. And in the country, the major producing area of calamansi is in this region, right here. That's region 4B. Actually, UPLB is located in this area right here. So we're located somewhere north in the country. Okay. And next, let's have pineapple. Pineapple is loved by Filipinos, but it's not that well produced because um, it's usually sold in the market at a higher price. And due to that reason, not a lot of Filipinos can afford this fruit. So it's usually sold as a delicacy or a luxury food for lots of us. Okay, and next slide please. And again, due to the absence of typhoons or very minimal typhoons that are passing in Mindanao, that's the reason why a lot of unique fruits or unique delicacies, or not delicacy, but unique crops are grown in that island. And next, we have coconut. What's coconut in Bahasa? Okay, we love coconut. I think Indonesia is the top producing country of coconut in the entire world. And we as Filipinos also produce lots of coconut as well. We use, in our country, we call it as, what do you call this? It's the tree of miracles or tree of life because every part or every portion of the coconut plant can be utilized or can be sold for money. And of course, as Filipinos, or sorry, as humans, we love money. You know, so who does not love money? Anyway, just kidding. But in the country was able to produce 14.49 million metric tons of coconut in 2016. And the top producing regions of coconut in our country is located here in near UPLB, near our university, and also in the lower portion of the country. The coconut industry of our country was recently ravaged by a pest, the coconut scale insect. It is believed to have come from Indonesia due to the importation of ornamental palms. The importation of ornamental palms, the ones that we use in the garden, it is believed that some coconut scale insects have attached themselves or have transported themselves using those ornamental palms, and that's the reason why the coconut scale insect was able to enter our country. And during those years, for 2010 to 2015, I think, the entire coconut industry of our country was somewhat paralyzed, but not entirely, but lots of coconut trees were damaged by that pest. Okay, so I think that's why it is important for us as students to know the different crops that are present in our neighboring countries, because we grow the same crop, and since we grow the same crop, we also have similar climate, we have similar climatic seasons or climatic variations, and that's the reason why it is important for us to look at a different perspective, and that different pests can move from one country to another. Our pests can not only move at short distances, but also at very large distances. That's why, as people of the Southeast Asia, we should be concerned, or also, I think it is also a good information for us to learn what are the crops grown in your country, what are the crops grown in our neighbors, and also the pests that are associated with them, in order to prepare us for globalization, or also for other activities that we as plant protect or we as practitioners of plant protections in the future might encounter. And next we also have sugarcane. A lot of Filipinos love sugar. We love sweet food, and that's the reason why in our country what we call this, the beverages that are usually possess high amount of sugar are taxed heavily, because lots of Filipinos have diabetes, and the government wants to restrict the use of sugar nowadays due to that problem. Next, okay. The major producing region of sugarcane in our country is this portion here. We have the island of Negros. That's the greenest here. This is the island of Negros, and in our country, that island is very famous for sugar. So when you say sugar, we call it asukal in Filipino. Can you say asukal? Asukal, yes. Asukal comes in many forms in our country. We have white sugar, brown sugar, black sugar, and again, lots of Filipinos love it. And the main producing area of our country is that island, the Negros Island. And next, let's have cassava. I believe Indonesians love cassava because in the place where we are staying, we're constantly served with cassava, either boiled cassava or cassava that has been cooked already into sweets or into this. And in the Philippines, cassava is actually not that produced at large amounts because only few Filipinos consume cassava, and sometimes in our food, cassava is used at minimal instances, actually. And the main producing areas of cassava is this portion, again, the north and the south. Okay, next we have onion. I think it's called bawang. Bawang here, bawang. Yes, bawang. Bawang. In the Philippines, bawang is garlic, which I think you call bawang puti, if I'm correct. Yes, bawang. But in your country, it's bawang. In our country, we call it sibuyas. Okay, sibuyas. We love sibuyas, although it makes us cry, right? Okay. In the Philippines, we only produce very small amount of onion, only 229.54 thousand metric tons in recent years. And the main producing area of onions, actually, can you look at the map? Not all provinces. Again, in the Philippines, we have 81 provinces. Not all of them are producing onions. Again, onions does not love a lot of rain, diba? Right? Onions does not thrive in rainy areas because the bulbs will rot due to the excessive amounts of water. And that's the reason why only a few areas in our country are producing onions. And in the Philippines, one of the major news every year in our country is the lack of onion. We actually are experiencing onion shortage in our country. Usually on the years of November, December, January, there's no onion. That's why the prices or the price of onion in our market usually rockets or skyrockets during those months. No? And usually the onion is at the same price. It's usually at the same price as some luxury crops. No? And that's the reason why in our country, it's one of our main problems. So that's why we have to intensify onion production in our country. And here's a short summary of the statistics that I have used in the previous slide. I just selected the not top 10, but just the common crops that we as Filipinos encounter. I believe Indonesians and Filipinos grow the same crop, but there's just some slight variations. But for our country, the main crops that we are producing in terms of volume of production are sugarcane, number one. Then we have rice, coconut, banana, and coffee. But for our country, the main... Now, for some ending remarks, okay, crops or growing crops in our country is usually a very profitable endeavor for a lot of Filipinos. A lot of Filipinos grow crops, different crops, depending on where they are located. And these crops provide lots of money for these farmers. But these are usually but the income are usually varies from crop to crop, of course. Horticultural crops are usually more expensive. Now in our country, that's usually more expensive. That's why the ones that are growing horticultural crops are usually more richer than the ones who are growing rice and corn. But anyway, more expensive. It's the same, okay. And the current threats that we as Filipinos experience for crop production are pest problems, are pest infestations. And these pest infestations are the main reason why a lot of farmers in the Philippines are experiencing loss of income, because whenever a single pest multiplies at a faster rate, they become a huge problem for the farm. And this results to huge economic losses for our farmers. Because whenever a single I believe in Indonesia in Indonesia you also have the same since we have the same crops, and also since we have similar crops, the pests that are found in our country are sometimes commonly found in your country as well. And the research interest or the research, what we call this, the research niche or the research opportunities for these types of pests that are present in both countries are usually large. We have, in our government, we have fundings that are coming from, of course, the taxes of the Filipino people. And these are usually used for funding researches that involve the common pest of these crops. And in our laboratory in UPLB, in IWEP UPLB, I belong in the insect ecology laboratory. And these are some of the insects that have been studied in the laboratory where I belong. We have the mango acid fly, Procontarinha frugivora. We also have the mango fruit fly, Bactrocera sp. Then we also have coconut scale insect, Aspidiotus destructor. Then the mango pulp weevil. These are some of the pests that are very damaging for Filipinos, usually mango. And these pests could be future threats to your country as well. Some of these pests, especially this one, is not found in your country. But since we are neighbors, so next slide please. Since we are neighbors, so here's a map of Philippines and Indonesia. Since we are neighbors, some of the pests that we are experiencing or that we are seeing in our country could be potential pests that could come in your country as well. They can come through importations or exportations. When these are not regulated properly, our problem could become your problem as well in the future. And the same way, your problem right now could be our problem in the future. So that's why, as plant protection students or as plant protection majors, we should be vigilant about the crops that are found in our country and our neighbors. And knowing what these crops are, and knowing what are the common pests that we have in both countries will be a fundamental knowledge that we can use in the future, in your future careers as plant protection majors. And that would be all. Terima kasih.
Question session. Kita langsung saja ya pertanyaan. Boleh in bahasa, boleh in English. Ya. Siapa yang mau bertanya pertama langsung dapat loh. Kita langsung saja ya. Okay, from here. Boleh in bahasa, boleh in English. Ya. Siapa yang mau bertanya pertama langsung dapat loh. Tes. Okay, from here.
Excuse me. Good afternoon, Mr. Enabor. First, my name is Safira, and I want to ask about golden rice. As I'm informed, golden rice was used for fight the deficiency problem in young people. I want to ask if the golden rice was cooked, how can the vitamin A is still in there and not loose?
So you're asking if golden rice, so when we cook the golden rice, the vitamin A will not be lost? Okay. Yes, it's the same way as in some other crops. Cooking golden rice does not remove the majority of the vitamin A that is found on the rice. So that's why after the cooking process, some of the vitamins will still be left in the rice. Yes, and it's actually the reason why rice was chosen as the one who will receive the beta-carotene gene is because rice is very accessible for lots of Filipinos. Again, rice or nasi is a very cheap crop and also a very fundamental food in our plates. So every day, 3 times a day, when we Filipinos eat, we have rice. We cannot sleep without rice. Every meal that does not have rice feels just like a merienda. What do you call merienda? Just like a snack or a quick snack. And that's why the intent in including vitamin A in rice or nasi is, I think, a very smart move because it is accessible for lots of Filipinos, even those below the economic bracket or those who are poor. So, yes, for your question, the vitamin A is still left in the rice, even if it is cooked already.
Thank you.
Okay. the vitamin A is still left in the rice.
Oke, Mbak Safira, selamat ya dapat kenang-kenangan. Nanyanya golden rice. Next, please. Oke, from far away. Oke, Mbak Safira, selamat ya dapat kenang-kenangan. Nanyanya golden rice. Next, please.
Oke, terima kasih atas kesempatannya. I'm very interested about the golden rice ya, Safira. Apa? And I was looking for a little info about the golden rice in Google. And golden rice is one of the GMO product ya. And as Mr. said earlier, this golden rice could be a solution to the condition of vitamin A deficiency. And what I want to ask is, what kind of organism that added to the rice DNA and the rice DNA so that it produced a golden color? And during the production, where there are any pests or disease that attack the golden rice? Thank you.
Okay. Yes. Thank you very much for the question. Golden rice is a GMO. It's genetically modified. So in order for the rice to produce vitamin A in its body and eventually have or deposit vitamin A on the grain, genetic engineering or biotechnological techniques has been prepared or has been done in order to incorporate the certain gene that is producing vitamin A. But as of the moment, sorry, I have no information regarding the organism where the gene come from. I don't have that information. I'm sorry. I'm very sorry. But nevertheless, in our country, a lot of some researchers are already proposing for projects that involve the pests that could possibly attack golden rice. Though some of the pests might just be the same as the ones attacking rice or the original rice or the rice which we are currently growing, such researches are still valid or are still warranted for future works. But for your question, I think it's the same types of pests that could possibly attack golden rice. But since it has been approved just recently, I think 2021 in our country, and it's still 2023, or we are still early in 2023. Such researches are still not being studied in our country. Thank you very much.
Okay, thank you. Two more.
Hi, I'm sorry, but according to my colleague here, the vitamin A gene came from daffodils. Am I correct? From daffodils. It's daffodil is the common name of the plant.
Flowers.
The flower it's from of course rice is also a flowering plant, but I'm talking about daffodils. It's it's an ornamental plant. So the gene that is producing vitamin A from that plant has been edited or has been taken away and incorporated into the genome of the rice. If I am correct. Am I right? Okay. My colleague says here that I'm okay.
Yeah.
Thank you very much for that question.
Oke, jadi teman-teman, rice tadi itu berasnya di GMO ya, jadi dimasukkan flowers. What kind of flowers?
Daffodil.
Dafus.
Thank you very much.
Ornamental plants yang memproduksi vitamin A, jadi disisipkan ke dalam padi, ya, sehingga dia bisa memproduksi vitamin A yang tinggi, ya. Jadi GMO sebenarnya bukan dari organisme yang lain, tapi dari tanaman juga, ya, from plants also. Ya, okay. One more question from here. First, I think we will give it to the men because two of them are women. Yeah, Bu Dewi. So the man in the middle. One more question. Yeah. First, I think we will give it to the men because two of them are women. Yeah, Bu Dewi. So the man in the middle.
Yeah. Thank you for the opportunity. My name is Noval, and I want to ask you about the production of the sugarcane. How was the agricultural system in Philippines that can reduce pest attack and disease uh, so as to produce a large amount of sugarcane? Thank you.
Okay, thank you very much for the question. Now, for sugar canes, the main pests that we have for sugarcane are usually weeds. We have lots of weeds when it comes to sugarcane production, and some of the insect pests that are attacking sugarcane in our country is usually the what do we call this? The rhinoceros beetle, the Oryctes rhinoceros. The Oryctes rhinoceros, the Coleoptera. It's a scarabate beetle. It is a root pest that is easily controlled by a lot of cultural and chemicals that are employed during the production. So that's the main insect, one of the main insects that is attacking sugarcane in the Philippines. But I believe we also have some plant diseases that are attacking it as well. But I'm not sure what is that specific disease. But for the insect pest, the main pest that I can mention is Oryctes rhinoceros, and it's easily controlled by some basic cultural management techniques, and also some the application of pesticide that is registered for sugarcane, and also registered for the control of that specific pest. Okay, thank you.
Now? Okay, thank you, Mr. Enaboro. Give a big applause for him. Thank you so much. Thank you. Just put it. Yeah. So a lot of plants actually we can say similar with Indonesia. Yeah, and you're talking about what it is. Onion, shallot. Yeah, actually in Indonesia we have the Philippine variety of shallot. I don't know you have it in your country or not, but its name is Philippine variety. I don't know whether it's come from Philippines or somewhere, but its name is Philippine. Yeah, the same condition with Indonesia. Sometimes it's when the what it is. We have a special day, then it's become also scary. We couldn't find this shallot in Indonesia also. The same condition. Okay, and then let's go to the next lecture today from Mr. Rejuso will talk about common insect pests in the Philippines. Okay, we read the CV. Mr. Rejuso is a Master of Science in Entomology and Horticulture as his minor, and graduated from Philippine University of the Philippines, Los Baños, and the Bachelor of Science in Agriculture and major in Entomology, also from Los Baños. And now his research interest is in biological control, insect ecology, and insect behavior. Okay, Mr. Rejuso, time is yours.
Thank you very much, ma'am. So, okay, thank you. Okay, so good afternoon once again, students of PTN1402. Am I right? Yes, okay. Salamat, Piang. Yes, I'm nearly perfecting it. And in our own language, magandang hapon. Okay, so good afternoon once again, I am PJ Rejuso, just like Sir Rex, an entomology assistant professor. And for today, I will discuss about this particular topic. I think there's a typographical error. My only focus now is common pests in the Philippines. Okay, but before I begin, I really like to commend this particular group of students because you are really listening to the lecture earlier. I know that you are diligent in your noting. Some are handwriting while others are using their laptop. Okay, so for today, for this specific time, I really like for you to listen because I will give a short exam later on. No, I cannot give it. Let's see. But some of the slides I will actually try to assess how you know the plant, your actual course, plant cleaning. Right, ma'am? Okay, now let us begin. Hello from the remote. Okay, next slide. Okay, here's the outline of my presentation. Actually, we strategized our presentation with Sir Rex and Sir Bethany for you to first be acquainted with the set of crops in our country, in the Philippines. Now we will dwell in with the actual pests. So how do we define pests initially? Then secondly, of course, we need to classify. How do we classify and group the set of insects that we actually encounter? And finally, some examples of the commonly associated pests in our country. We have a very abundant list of pests, but as of now, I will just list some commonly associated pests. Okay, next slide, sir. Okay, so how do we define pests? So what do we think about when we hear the word pest? Actually, the term pest, next slide, is just a perception of human. It is relative to our own assessment of a particular organism. A pest is only considered a pest if it negatively affects our own selves. Next slide. Okay. It creates annoyance, and finally, to put it simply, it either hurt, damages us ourselves, the crops, livestock, or our properties. In our discipline per se, our main focus are those organisms directly affecting our crops or our livestock. Those are related to agricultural discipline. Okay? Is it clear? Hey, okay. Next slide. So now let us talk about how do we classify pests. So there are several aspects that we can classify the pests. We can either look at their biology, morphology, or direct impact towards our crop. Firstly, we can classify them based on their feeding habits. Number one, when their mouth part, I know you are very familiar with the mouth part of the organism, our insect. Next slide. When the mouth part of the insect is chewing or mandibulate, their actual damage will be defoliation. Next slide. So, in the case of their piercing, sucking mouth part, some of our insect has those particular mouth parts. Their damage will be defoliation. And finally, these are my favorite. Some of immature of insect serve as a pest, and they cause damage by their boring motion. Okay, now let us talk about classification of pests based on the host plant. Some of the host plants listed here are present in our country. Firstly, some of the pests attacks cereals or the grain crops. So what are the example of grain crops? Grain crops, cereals. Very good, rice. I hear the word rice. Rice, grain, and corn. Wheat and corn. Then we also have this particular crop. What is this? I think this is not very clear. So some of the pests attack our horticultural, or in this case, fruit crops. Can you click? Yeah, fruit crops. Okay, what is this crop? Yeah, very good. Very good. Okay. Some of our pests attacks solanaceous crops. What are the example of solanaceous crops? Yes, very good. I think I heard eggplant. Eggplant, tomato. Then some of our pests attack plantation crops. What are the example of plantation crops? Yan, coffee, cacao. And finally, we also have those pests attacking legumes, peanuts, soybean, mung bean. And finally, some of them attack cruciferous, the leafy vegetable crops. Okay, next. So, insect pest classification can also be considered as in temporal, in spatial aspect. Where do they specifically attack our target crop? So some of the pests attack the vegetative parts, the leaves. Some of them attack the stem. Those some of them are those boring insects. Then we also have those attacking the root organs. So what is this organism? Okay, we'll talk about it later. And some of them attacks flowers. So what is this insect? Are you familiar with this? Yes, very good. I really like this class. Those are thrips. Okay, and finally, some of them attack the actual fruit organ. Okay? Okay, now that we talk about the spatial aspect of our plant, now let us talk about the temporal or the phenological stage upon which they are attacking. So what crop is this? No, it's actually, it's corn. Very good. You really are agriculturist. Okay, Of course, in the growth stage of corn, we can specifically divide it into two, the vegetative and the reproductive. So those attacking at early seedling stage, where in the stem is really succulent, those are the praising sucking, the aphids, the plant hoppers. But when our corn plant transition to middle to late vegetative stage, now we can observe the pollinators, the fall armyworm, the Asiatic horned borer. And finally, when it transitions to its most productive stage, the reproductive stage, now we can see those attacking the panicle, silk, and ear feeders. And of course, when we talk about different parts, we should be able to differentiate those attacking the annual and the perennial. So what crop is this? What is this crop? Oh, very good. It is mango. Okay, the difference in perennial crops, as we all know, their growth stages is repetitive. Okay, next. What is this crop? Okay, since they are repetitive, their vegetative and reproductive stage is continuously happening all throughout the life cycle of our crop. So during its vegetative stage, there are those attacking the leaf and shoot feeder. There are those considered as leaf and shoot feeder. So what are the example of these? The tip borers, the longhorn beetle. Now, when it transitions to flowering stage, now we can observe those feeding on the flower, of course, such as the mango flower hoppers or mango leaf hopper in our case, the thrips. And finally, those once in transition to fruit developmental stage, those there comes those serve as fruit borers and feeders. And finally, ultimately, our most commonly associated classification is based on their actual damage. So in the case of damage, we can classify our description into two, those that we consider as the symptom and the sign. So are you familiar with that? Yes, this is very good class. Okay, what is the difference? Okay, I will just state. So the symptom, so for example, what is this? Is this a symptom or a sign? Let's see. It's lip rolling. Okay. So actually, the difference between a symptom and a symptom is the actual manifestation of the injury towards our crop. Okay? So while the sign is where you actually observe the causal organism. When you see the faces, the actual organism, it's exoskeleton. So when assessing damage, you should be able to recognize these particular evidences. Okay? Since we are considered as plant doctors, right? We are plant doctors. Our target patient is not speaking, so we should be able to be Sherlock Holmes. We should be able to interpret these particular symptoms and signs. Okay? Some of the key signs and symptoms that we recognize, of course, when we have those defoliator, the feeding on the leaves. We also have those leaf rollers, the stunted growth. So usually, when you have this particular symptom, our key pest is attacking our root organ. We also have pathogen transmission. So what is this crop? Oh, very good rice. So in this particular image, this particular rice is exhibiting symptom of tungro disease. And finally, those attacking our fruits, where you can specifically see the damages on the organ. Okay? Next slide. So now let us go to the actual example of our commonly associated pests in our crops. Okay? Okay. In recognizing once again the target pests of our concern, we should be able to at least recognize the key characters of our insect. In this case, target organism. Firstly, its morphological characters, and secondly, its biology. Okay? Morphological aspect in the basis of their feeding habits. Of course, this will tell you how will they potentially damage our crop. And there is their biology. So what aspect of biology are we talking about? Their life cycle. Because some of our target insects are more susceptible at a particular stage. Okay? Is that good? Yes, very good. By remote, okay? Yes. Okay. Next slide. Okay, firstly, we have our what we call as salagubang locally. June beetle, scientifically named as Leopolis irorata. Under order, what order is this? Oh, very good. Nice. Coleoptera. Okay. So this particular... Yeah, hey, Coleoptera. You're right. Very good. Everyone get keychains. Okay. This particular insect is considered as pests in our annual crops, such as what mentioned by Sir Rex, the sugarcane, but more importantly, towards our rice and corn. Okay? In terms of its feeding habit, the one that is really detrimental towards our crops are its what stage? Its larval stage. Okay? The larva feeds on the root areas of the developing crops. Our crops is more prone to this attack during their earlier stages. Okay? So in terms of their damage, how can you tell whether this particular pest is present in our crop? Their feeding habits causes stunting, nutritional deficiencies, and eventually stand loss. Eventually the death of our crop. Okay? So based on their feeding habits, damage, biology, how can we simply control this particular pest? So their susceptible and damaging pest is their larval position underground. So through good cultural management, soil preparation, you can control this particular pest. Okay? So what pest is this? June beetle. What is your local term for June beetle? So why is it called June beetle? So ideally, the adult of this is, at least in our country, is prevalent during the month of June. Okay. What is your local term for June beetle? Toy beetle. Because toy beetle also because when we were young, around 2 years ago, we used to play with this. We used to play with this particular pest. Are you also playing with this particular insect? Oh no, you're okay. Yes, ma'am. Next slide. Okay, our next pest is several stem borer species. At least my focus here is the striped stem borer, Chilosuppressalis, under order. Next slide. Oh, very good. This class is really good. Under order Lepidoptera. So remember, in this particular insect order, the damaging stage is once again, the larval stage. So in this case, the immature feeds inside the stem of the rice. Okay? And their overall damage is their feeding habit causes younger leaves and growing point to wither and empty panicles. So take note, there are two distinguished symptoms that can be manifested as caused by this particular pest. The dead heart during earlier stages of rice and whitehead during the reproductive stage of rice. Okay, next slide. So what is this? Is this a sign or symptom? Symptom. Okay, it's actually symptom. Very good. Okay, these are pictures exhibiting dead heart and whitehead. So why is it called dead heart? Because remember the parts of our rice. So the central pillar is actually brown or dead. So because in the heart it is the center of our body, right? So in this case, the central pillar is dead, caused by the feeding or the boring feeding habit of our insect pests. So remember, when our insect is considered a stem borer, they are disrupting the vascular system of our crop, making them dead eventually. So while, once again, in the case of our stem borer, where they attack at a much later stage, when the rice reaches reproductive stage, it will incur this particular symptom, whitehead. So why is it called whitehead? Because they, very good, yes, I know you are correct. The panicle is colored white. So what is the usual color of our rice panicles? Yellow, golden, right? So in this case, it is empty, so that is why the panicles are really colored white. Okay? So usually this particular pest is controlled by application of insecticides. Next slide. Okay, now we have our GLH, what we call as green leaf hopper. Nepothetics virescent, also an important pest of rice. Under order, very good. This class is really good in taxonomy. Really? No. No, I believe in you. You're not cheating. Okay? So in this part, no, now we have different case from the two previous pests. So this particular pest is undergoing different mode of development. So both its immature and adult can be considered as pests. So nymphs and adults feed on plant sap using their piercing sucking mouth parts to pierce through the leaves and stem of the developing rice. So overall, their damage is eventually translated to upper leaves whitening, young plants stop and grow, and more importantly, in our case, it is more important as vector of this particular virus. So Tungra virus causing what disease? Oh, very good. Tungra disease. Okay. So this particular pest is actually controlled by chemical control tacticals. Next slide. So we also have red ant. So Solenopsis geminata under order. Oh, very good. I recognize Hymenoptera. Okay. So this particular pest is only considered as pest when our farm is practicing an upland and direct seeding method. Because remember the habit of ants, right? They really like starchy material. So they will collect and eat these particular materials when we are practicing direct seeding, such as the rice seeds, right? So pests of broadcasted upland rice where they will feed on broadcasted seeds before they germinate. So eventually translating to patchy distribution of broadcasted rice upon germination. So because there will be no germination since our ants already collected this particular plant materials. Okay? So how do we manage this particular pest? You can manage it by practicing lowland condition of rice or practicing transplantation. Okay? Next slide. So I believe this is your most concerning pest in rice, right? So we have our brown plant hopper, BPA simply, Nila parvata lugens, under order, Miptera delphasi. Okay? Same case with our green leaf hopper. Both its nymph and adult feed on the plant sap using their piercing sucking mouth part. So their overall damage is translated to desiccation of the total plant area. Okay? However, this particular pest is known to aggregate. So the aggregation actually causes the burning-like symptom, what we call as hopper burn. Okay? So additionally, they also are also known to transmit grassy and ragged stunt virus. So control still via chemical control. Next slide. And of course, in our country, this is the most concerning pest, Scotinopara sp, either coornata or other species. Rice black bug. Do you also have this particular pest? Yes, of course. I know it's distribution. Yes, you also have this. Under order, Hemiptera. Still, based on its mouth part and biology, its damage is still translated to overall plant desiccation. Immature adults aggregate. This is important. It is important for you to know where they are located. They usually aggregate near the base of the rice. So damage, yellowing of leaves, stunting, reduced tillering, and even death. Okay? So control through chemical control tactic. Next. Okay, now we transfer to different crop. So we have this particular pest, Ostrinia furnicalis, Asiatic corn borer, under order Lepidoptera. Okay, so what does it target? What particular crop? Okay, very good, corn, of course. Okay, so prior to the introduction of BT corn in at least in our country, this is the most prominent pest of our corn crop. Okay, so the larva bore holes on leaves, stem, ears, and stalk of corn plants. Actually, this particular pest attack all parts of our corn plant except the roots. Okay, so its damage is manifested by small dark color entry holes, stalk rot symptom, and eventual stalk lodging. So once again, this particular pest is now easily controlled using the BT corn varieties as planting material. Next slide. Because nowadays, our main pest of concern in corn is this particular pest, the fall armyworm. So I believe this is also your pest, right? But in different crop. What particular crop? Acorn also. Okay. So this particular pest is considered as polyphagous. What do you mean by that? So they target several host resources. They not only target corn, they are also known to feed on onion and several solanaceous crops. So their feeding habit, larvae can feed on all plant matters at wide aggregation. That is why they are called an army. They are attacking in aggregation, eventually really translating to the overall crop failure. Okay? So damage yan. Total vegetative part, plant part destruction. So nowadays, as we mentioned earlier, this is the actual key pest in our country. So several research studies, at least in our university, is really focused on its management, either through chemical control tactic, pheromone-based approach, and even biological control. Next slide. So what is this? So we, as mentioned by Sir Rex earlier, once again, the coconut rhinoceros beetle. So what is its target host plant? Yes, correct. I believe that is coconut, right? That is coconut, right? Yes, that is coconut. Okay, so Erectus rhinoceros. I believe I saw this particular pest yesterday. Okay? No, actually it's elephant beetle. Okay, on this particular pest, both its immature and adult is pest towards our target crop of coconut. Okay? But in their case, the larva and the adult have different target plant organ. The larva feed on the plant part roots, while the adult feeds on the vegetative part, the leaf, the leaf. Okay, its damage is translated to root damage when we are considering the larva, while the adult born and open leaf. So it is easily observed in the field because once you observe leaf of coconut palm, you will observe this particular damage, the triangular pattern in the leaflet. Okay, so control, we are practicing chemical control on this particular pest. And also the release of metarhizium bioconjugate. Next. Okay, this is what I'm most interested. Have you heard of this particular pest? Coconut scale insect. Really? Yes. Earlier, this is mentioned by Sir Rex. Aspidiotus sp. Or because there are two species known, at least in our country, to cause devastation in our coconut industry. Aspidiotus destructor and Aspidiotus pigilus. Okay. So I think you are familiar with this. So it's one click. Okay, it is under suborder Sternorhyncha, or those hemiptera classified or characterized by being less mobile or even sedentary. Okay, so its feeding habit, both nymph and adult, suck plant sap in leaf section. So in our country, once again, as implied by Sir Rex, it is one of major pest during 2010s to 2014s because it is really devastated our coconut plantation, starting from our main island of Luzon and to some regions in the Visayas and even Mindanao. Actually, currently, it is still a pest in some regions of our country. Controlling this particular pest is really tricky. So why is that? Because it is attacking coconut and it is attacking the leaflet of coconut. So as you all know, coconut has really high elevation, large height. So we cannot really release our insecticide basically. Okay? So its damage is translated to plant part desiccation. Of course, the leaflet and reduction in photosynthetic activity. Okay? So actually, this particular pest is controlled through several actions by the government and of course by the experts in our field. So it is controlled through, once again, insecticide application. But insecticide application, that is not normal. When you are applying insecticide in here, you should be using a high-powered sprayer, such as those used in the fire trucks, once again, to eventually reach the high elevation of leaflet. Another method of application is trunk injection. So you are releasing the insecticide via its vascular tissues. And of course, several practices can be also done. Pruning. When you are observing the initial symptom of this particular pest, you can start pruning the leaflets. And actually, this particular pest is really controlled, quote unquote, and its population is really reduced. Can you guess how? By a natural calamity, by a typhoon. Okay, because I believe it is Glenda around 2014 when this particular typhoon passed through the Philippines. It really devastated all of its surrounding, including the coconut. So their leaflet, the leaves of the coconut are really destroyed together with this particular pest. Okay, but as I mentioned earlier, this particular pest is still devastating some other parts of our country. Okay, just a note, what mentioned, there are actually two hypotheses in the introduction of this particular pest. One, introduction, as mentioned by Sir Rex, from neighboring country. Okay. However, there is also a paradigm or worldview that this particular pest is actually present locally in our country. However, a particular climatic weather condition just promotes its population rise. Okay, so this is coconut scale insect. Next slide. Okay, so now we go to vegetable crops. Okay, I will be needing only 5 minutes. Okay, so we have eggplant fruit and shoot borer, Leisonades orbonalis. So still Lepidoptera. This one is targeting. What do you think? What crop? Eggplant. Yes, very good. Okay, this is the larva bore and feeds on shoot, flower, or fruit. Okay, this particular key pest is the reason why our eggplant cultivation is requiring a lot of chemical application. Okay, larva cause shoot, flower, or fruit wilting and eventually drop off. So as I mentioned, this particular pest is controlled massively through chemical control tactic. But nowadays, there are commonly associated biocon agents and even utilization of GMO crops to control this particular pest. Next, just few more slides. Actually, this is not fruit fly. Typo error. This is what Sir Rick referred earlier as Bingo sessile fly, Procontaria frugivora. So this particular pest is attacking our mango fruits. This is not edited. The adult is the one ovipositing on the mango fruits. Oh, it's right. While the larva is the one feeding on the exocarp or the peel of our crop. Manifesting as gall. Do you know gall? Gall. Just deviation from the normal appearance of the exocarp. So damage, gall formation in the exocarp of fruit, eventually turning into exit hole, what we refer as curricle. Okay, so this is mainly managed through chemical application. Next. Yay, the actual fruit fly. Okay, this is the actual fruit fly, okay? That is just a test, okay? So, under species Bactrocera dorsalis. This particular pest is attacking what crop? Yay, the actual fruit fly. Okay, so very good, mango. But remember, this particular pest is also considered as Pholipagus. This is attacking several crops. From fruit crops, the guava, breadfruit, and even solanaceous crops, tomatoes. It is known to attack tomatoes. So adult appears on maturing fruit while the larva feeds inside the fruits. So this particular pest is considered as quarantine concerning pest. This insect is actually not reducing the production, but considered as a threat towards importation and export of a particular crop, in this case, mango. So that is why it is still important to control this particular pest. So adult causes quality reduction, larva infestation results to fruit deterioration. So in our country, this is mainly controlled through mango bagging. And there are also known chemical chemical insecticide. against this particular pest. Mainly controlled through mango. Next slide. Okay, now we are done with some insect pests. I will just like to introduce some of the pests not considered as insect, such as this particular pest, golden apple snail. Okay, Powasea caniculata. So a mollusk snail. So this particular pest is targeting rice plantations. Feeds on the rice young plants using their mouthpart known as radula. And they prepare soft and young plant parts rather than the maturing one. So damage that of young rice plants, older plants tend to tolerate this particular feeding. So same case with some of our pests. This particular pest is also an introduced one. So golden apple snail or Powasea caniculata is actually imported from South America, from Argentina during the 1980s. So initially there is a good objective for the introduction of this particular organism. That is to serve as protein substitute. However, our government that time overlooked the fact that this particular organism can also target rice plants. So control, proper water management. Next. Okay, we also have vertebrate pests. So I believe you have expert in vertebrate pests here, right? So I will not dwell in much to this. I believe you are really familiar with the impact of vertebrate, or in this case, the rats. Some of our rat species. Next. So their damage is really distinguished because they have chewing mouth parts just like us, actual tooth or teeth. So their damage is exhibited by the missing parts or some of the cuttings in the rice plantation. So it is easily observed when they are really infesting because there is an uneven elevation of rice plants. Yeah, missing crop, irregular stump cutting. And finally, some bird species. Okay? So I believe you know how to control this particular pest. You are using nets, right? Right? Okay, so in terms of their damage, they cause damage because they really like ripening or the actual grain. Okay? Chew rice grains and can cause whitehead or unpeeled panicles. So they can either be, they are prominent during the ripening stage of our rice. Okay? So controlling mechanical tactics, using of traps, installation of barriers. Okay, just like what you are currently doing. Okay, so to summarize, so those are just some of the sample pests, key pests in our country, but there are still more. But we do not have much time. We only have 10 minutes. So in summary, insects affect agriculture negatively or positively depending on its identity. What do I mean by its identity? Based on its biology and morphology. So proper identification of insect is integral component of successful pest management. So remember, the initial stage of pest management is recognizing our enemy. So once you recognize the actual causal organism of our pest problem, now you can proceed with the actual pest management program. Okay, so that is why it is important for us, agricultural students or plant protection students, to be able to recognize the evidences, the signs and the symptoms of a particular damaging insect pest. Okay, and that is all. Terima kasih.
Okay. Question. 1, 2, 3. First. Yeah, yeah. Okay.
Easy.
Question.
Parang 20 minutes lang.
1, 2, 3. First. Yeah, yeah.
Okay. Sorry if my English isn't good. I will ask. From what I heard, from presentation earlier, Indonesia and Philippines have same main food. It is rice, right? Nah, in Indonesia, one of the best of rice is rats, like in the presentation. Nah, and we we have an old culture named gropiokan. uh, which is still Indonesian farmer do to control the rat population. For your information, Gropiokan is an culture that people gather to catch and searching for rat nest to control population. What I want to ask is, in the Philippines, there is an old culture like that or no? Thank you.
Okay. Thank you very much. So I believe, if I interpret it correctly, there is your program in your country where the farmers are encouraged to collect, right, the rats.
Right.
So actually, it is the same in our country. So some of our local communities are encouraging the collection of this particular pest in order for them to be self-sustaining. However, some of our areas actually do not encourage this practice because take note, this particular collection has rewards, right? Same with you. Rewards once you collect an rat. There is a accompanying reward when you surrender this particular rat. Because some of our farmers are really wise, they are actually culturing the rats themselves for them to be able to get the money as their reward. But some of our communities really practice this particular program for them to be encouraged to be self-sustaining farms.
Okay, so you have the same.
Yes,
In
ma'am.
Indonesia, we call it grobiokan.
Grobio.
Grobiokan.
Grobio.
So calling the rat together in the farmers
Yes, ma'am.
and then collect it but
Collection.
sometimes this kepala desa also give money to them.
Yes. Same with us, ma'am.
The leader of the village.
Yes,
So give
and
them-
they have groups to collect this
Yes,
particular
yes.
pest.
Okay, the same with your country. Indonesia sama Philippines.
Of course, we have same faces, same culture.
Same culture.
Same culture.
Okay, thank you.
Thank you very much. They have groups to collect this particular pest.
Indonesia sama Philippines.
Of course, we have same faces, same culture.
Same culture.
Same culture.
Okay, thank you.
Thank you very much. Kaya mo yun. 1 hour pa.
Okay,
Go ahead,
thank
ma'am.
you. Now I want to ask you, but explain just now, what pest cause the most losses in agriculture in Philippines, especially in food crops?
1 hour pa.
Okay, thank you. Now I want to ask you,
On the fruit crops.
but
Okay,
explain just now,
fruit crops.
what
Okay.
pest cause
So actually, once again, there is thank you for the question once again. To begin with, so at least in our focus fruit crops nowadays, we are focusing on those high value crops such as the mango. So in the case of mango, the most concerning pest is what we earlier discussed, the mango sessile fly or the mango sessile midge, the Procontaria frugivora. Because this particular pest is really attacking our mango crop at its most vulnerable stage when it is earlier, when it is at its fruitlet stage. This particular pest, whenever it is reaching outbreak population, there is an actual 100% loss in the production of several mango farmers. And currently, there are several news outlets informing us of the outbreak of this particular pest. And our farmers are really trying to only just to look for a solution for this particular pest problem. The mango sessile fly.
Thank you. So you mentioned about kurikong.
Korikong, ma'am.
Yeah.
That is our local term for sessile fly.
Okay, thank you. So masalah utama Procontarina
Frugivora.
frugivora. Yeah.
I believe, ma'am, as mentioned yesterday, you also have this particular genus, at least genus.
Yeah, yes.
But in your case, this particular species, Procontaria mangifera, is
Procontarina.
only attacking the leaves.
The leaves.
Not the
Yeah,
fruit.
yeah,
Your
not
seed,
the
ma'am.
fruit.
Your
Yeah,
seed.
okay.
The-
So kita
current-
juga punya tapi hanya menyerang daun. Tapi
Currently,
tidak,
there is no record of this particular pest in your country.
Yes, yes. Okay. Okay, thank you. Next. Here or some kita juga punya tapi the back. No.
Questions? Go ahead, students.
Okay, thank you. Next.
Ayun. Ayun, namimigay na si ma'am. Hindi eh. Go ahead, students. Brochures.
Yes.
You can read our brochures.
Okay,
Go
sir. Thank you for the time. I want to ask you two questions. The first, you have explained a lot of pests, and I heard that the control is using chemicals. So, I want to ask you, the apa? You have explained a lot of pests. Biological or environmentally friendly control, such biological pesticides or other. The second, please explain for us integrated control to control that pest that you have explained. Thank you.
Okay, I will answer first the first question. Thank you very much for that question initially. Okay, so I believe if I am interpreting it correctly, you are commenting on the fact that most of our control tactic envisioned in this particular presentation is about chemical control, right? So as we mentioned earlier, as we implied in our most presentation, the control, the conventional control, at least I believe it's the same in your country. The conventional control tactic against any pest is really utilizing pesticides, either insecticide, fungicide, or herbicide. So it is really hard. Actually, I'm a biological control tactic. Ano ba yan? Purveyor. Because I really like to have this particular alternative towards chemical control. However, it is really hard to set this particular envision for our farmers because they really want this particular quick action towards a particular pest problem. Okay? And compared to alternatives, this is more chemical control tactics is really more affordable for them. It is more convenient for them to be utilized. And your second question, I'm sorry, I did not actually get it. Right. This is more chemical control tactics is really more affordable for them. It is more IPM. IPM. Okay, IPM of single pest. Okay. Is it right? IPM of a single pest. Okay. So I will focus on on my favorite insect pest, the coconut scale insect. Okay. So in this particular pest, once again, the main focus during early 2014 is really utilization of chemical control. Okay. So, because as I believe you all know, in order for us to really reduce the population of a particular pest is using a really effective chemical control. So during this really outbreak phase, they are really looking for an effective way of reaching this particular active ingredients towards our target pest. So the main approach is chemical control. However, they are also encouraged to practice cultural management, such as pruning. So once they observe starting population or the key symptom, the yellowing of the leaflet, our farmers are encouraged to practice pruning to reduce starting population. Then sanitation, and eventually, once you found out the proper biocon agent against this particular pest, it is also encouraged to release these particular parasitoids. Then sanitation, and eventually, once you
Jadi Mr. Regiso menceritakan tentang bagaimana yang scale pada coconut, bagaimana mengendalikan secara integratif, ya. Jadi bukan hanya chemicals. Tadi yang disebutkan itu beliau menyebutkan tentang chemicals itu praktis yang dilakukan oleh kemungkinan sebenarnya bukan petani tapi lebih ke tingkat pedagang. Jadi post harvest treatmentnya banyak yang dilakukan seperti itu. Tentu idealnya because we have plant protection, so kita sedang berusaha how to find the environmentally friendly method to control the disease, pests in the field. Yeah, so this is our future vision. Yeah, of course. The same with our friend from Philippines. He just mentioned about the practice now, what the farmers or what the trader do to treat the post-harvest pest in the Philippines. Yeah, I think like that.
Perfect,
Yeah.
ma'am.
Yeah.
Exactly.
Okay, so the real condition now. But then you, the next generation, have to find. You still young also. I'm in the middle now.
Exactly.
To find the way to treat our plants in the right way for our future. Yep. Okay. The last for you.
Okay, good afternoon. Let me introduce myself. My name Tere. I wanna ask Mr. Rejuso. I think we have a similar pest crops at that slide, like Schitinopara, Nepotetics, and another. I wanna ask, is there a biological control, another biological control beside metarhizium? Because is there a biological control,
Ano po?
another biological control beside metarhizium?
Biological control. Meron nga ba? Wait lang. Thank you very much for that very analytical question. So actually, nowadays, the most prominent, at least in our country, to be utilized against this particular pest, the Scotinopara, the rice black bug, is the what you mentioned as Metarhizium anisoplay, I believe. So, however, there are several species of Metarhizium, right, that are currently being assessed for their bioefficacy. But throughout the experimentation, of at least initial experimentation, they are now finding this particular species to be at least somewhat ideal for them to be utilized at field condition. But mainly the focus of the control, at least biological control tactic approach against this particular pest are utilization of entomopathogenic organisms. Did I answer your question, Elaine? Oh.
Thank you, and thank you, Mr. Rejuso, for the nice presentation, broadening our knowledge and remembering us again what the pest on the plants. Okay, thank you again. I think we need to drink. Thank you so much. And then next, from Mr. Macasero. He was talking about disease diagnosis in Philippines. Time is yours.
Yeah. Actually, this is not just the Philippines, so I will be discussing disease diagnosis in general. How do you diagnose different diseases?
I'm sorry, I have to read your CV first.
Actually, this is not just the Philippines, so I will be discussing disease diagnosis in general.
Okay, sorry. Mr. Macaruso is a Master of Science from Philippines, from Los Baños in plant pathology. So you have no minor like him? No minor?
Actually, we have minor, but we don't usually have.
Oh, okay. And Bachelor of Science in Agriculture, major in plant pathology, also from University of the Philippines, Los Baños. Okay, time is
We
yours.
have minor, but we don't. Okay, thank you. Okay, so it's not right written there, but my minor is in agronomy. Okay. Yes. So I will be discussing the procedures on how you diagnose, the importance of diagnosis, because just like the insect pests, they are detrimental to the growth of the plant, but the problem is you cannot see the pathogens. You can only see the disease, right? Who has seen a fungi up close? Without the microscope. Who can see fungi without the microscope? No, diba? No. Yeah, for the pathogens, you cannot see them with your naked eye. So for the diagnosis of different pathogens or different diseases, the process is a bit different from those of the insects. For insects, you just look at them, identify them, and then you can know what the insect is or what is the causal organism. But for pathogens, you need a proper set of procedures before you can identify these different pathogens. Next slide. So, first of all, plant disease diagnosis, the definition is the identification of the disease of a plant and determination of its causal organism. So this determination of the causal organism is through their signs and symptoms. So Sir P.J. Rehuso already mentioned earlier about the different signs and symptoms, the definition of both signs and symptoms. So why do we use this method? For example, what is the difference between a tungro-infected field and a nitrogen-deficient field? Can you identify? Who has seen a tungro-infested field, infected field? Tungro. Have you seen tungro before? Raise your hands. Not yet? What's the local name for tungro? Tungro still. Okay. So I think you already see, because you are the crop protection students, I think you already see, but I also have pictures later on. So we use disease diagnosis because we want to know what is the exact identity of that disease. Because for diseases, it might look like this one, but it's actually caused by a different pathogens. So later on, I will be discussing the importance of plant disease diagnosis. Next slide, please. So I think Sir PJ already mentioned the differences between symptoms and signs. Symptoms is the manifestation of the plant. Anything that happened in the plant or anything that is altered within the plant, it's called a symptom. But those that are visible manifestation of the pathogen or the presence of the pathogen, we call that the sign. Next slide, please. So these are symptoms. So they are alterations of the plant. So what happened in this case? The symptom is bunchy top. So the disease is banana bunchy top virus. What happened to the plant? It became stunted. Okay? So that's a symptom. Stunting is a symptom. So what is this one? So the disease is banana bunchy top virus. This is citrus canker. So there is canker lesions here. So canker is a symptom. And then lastly, what is this one? The crop is cabbage. And what happened to the roots of the cabbage? There is a club root of cabbage. Okay, so clubbing of the roots. So those happened on the plant itself. So that is a symptom. So next slide. These are signs. So for diseases, we have different signs compared to the insects. So we have signs for viruses. We don't have signs for viruses because we cannot see the viruses. Okay. We have signs for fungi, for bacteria, and for what's this one? Nematodes. Okay. This one is, what is this one? Actually, this is not fungi. This is a fungi-like oomycete. This is Peronosclerospora philippinensis or downy mildew of corn. Okay? That's why I use that slide. It's not that easy to understand. This one, oh, okay. Okay, you have two different Peronosclerospora. Okay, so this one is a fungi. So actually, I'm not familiar with this one because this crop is not very, it's a pumpkin. We don't grow pumpkin that much in the Philippines. And this one is, what is this one? This one? There is, what is this one? This is, it looks like dew, but the color is different. So actually this is bacterial ooze. Okay? Oh, I know, you know bacterial ooze. But when it's in the picture, maybe because the picture is also blurred, but that is bacterial ooze. So for bacteria, the only sign that you can see is bacterial ooze. For fungi, there are a lot of signs that you can see. You can see the mycelia. You can see if there are no spores, what will you see? For rhizoctonia, what will you see? You will see the sclerosal bodies. Okay, that's why I did not say sclerotinium because you will know it's sclerotia. I said rhizoctonia. Also, you can also see the spores. The spores are also the signs, but it's microscopic. So there are macroscopic signs. Microscopic meaning you need the microscope to see these signs. But for macroscopic, you can just use your naked eye. So this is the only sign for bacteria, the bacterial ooze. For nematodes, what are the different signs? These are, what is this? This is the female of the cyst nematode, or maybe it's the root knot nematode, right? But you can also see the male or the juvenile. Sometimes you can, in some species, you can see them without using the dissecting microscope. This one, you can also see the egg mass outside of the roots in most cases. The jelly-like substance outside of the roots. Next slide, please. So what's the importance of disease diagnosis? Do I need to be here for the picture? I like to walk while doing my lectures. Okay, so what's the importance of disease diagnosis? So the proper identification of the causal organism leads to proper management. So we have a lot of pathogen groups, and within those pathogen groups, we have different causal organisms. For example, what are the different pathogen groups that we have? We have fungi, like mycetes, bacteria, phytoplasma, nematode, virus, viroids. What else? What else? Actually, we still consider phanerogams or the parasitic flowering plants as pathogens because they cause symptoms on the plant. Okay? So there are a lot of causal organisms. There are a lot of pathogen groups, and each of these different pathogen groups have different management. Later on, when we go to the rice diseases, I will be mentioning how you manage these different pathogens. Okay, so I'll only be discussing rice because that's actually my specialization. I've been working with rice for more than 10 years, actually. So I'm a bit old. Yeah. Okay. Then another one is proper management means less cost or losses. Okay. So what do I mean by that? For example, let's use a chemical application. For example, if you use a bactericide against a nematode, what will happen? Will you control the pathogen? No. So you are given additional cost because you used the bactericide against a pathogen, against the nematode. So you're given additional cost, and you will not be controlling that pathogen or that disease. So what will happen to your crop? You will have additional losses because for diseases, for pests, every added day or weeks or months in your management, the pathogen or the pests are there in the crop and you are failing to manage them, there are additional losses. Okay? If for example, your harvest should be around 10,000 tons per hectare, and then that losses gave you around 6,000 tons per hectare. So you lost how many tons per hectare? 10 to 6. You lost 4 tons per hectare. And if you compute it, how much is rice here in the Philippines, in Indonesia per kilo? Paddy rice. Ma'am? Yes, ma'am. 4 tons per hectare. And if you compute it, how much is rice here in the Philippines, in Indonesia per kilo? Paddy rice. Ma'am? Yes, ma'am. Okay, and then you multiply it to 6,000 because one ton is equivalent to 1,000 kilos. So that's the amount of losses you have, right, ma'am? So actually, it's part of IPM management. The correct management is always part of the IPM management. So next slide, please. So in disease diagnosis, we have 2 general methods. First is the direct inspection. The next one is the deferred inspection. So next slide. So direct inspection is the direct examination of the diseased plant. So without incubation. Okay, no incubation needed. So you look at the plant, you look at their symptoms, and then you identify which pathogen or what disease there is. Okay? But the problem is, in that one, it is performed by an expert. Okay, for example, for me, I've been working with rice for a very long time. I already know all of the different diseases of rice in the Philippines. Okay, in the Philippines. If someone in the Philippines brings a sample to me for identification, I can identify that disease already. Okay? So that's the limitations of direct inspection. Because again, what will happen if the identification is wrong? You might give the wrong management recommendation to the farmer. So this one is always done by an expert. So next slide, please. Deferred inspection is the examination of the causal organism after incubation. Okay, so later on, I will be showing you different incubation methods for fungi, for bacteria, for nematodes, and then a different one for viruses. Okay. So this is especially useful for when unfamiliar with disease or there is a disease complex. For example, you see a lot of diseases or a lot of symptoms, and you do not know what is the original causal organism. Okay, so it happens a lot when it comes to the field. In the field, you will always see a lot of diseases, not just diseases, also pests. Okay, insects. Okay, and all of those will cause something on the plant. So you need to identify each of the different ones to have an integrated management recommendation for all of those crops. So you don't recommend just for if the field has bacterial leaf blight, has the green leaf hopper, and has the sheath blight, you will not recommend only for bacterial leaf blight. You will always recommend for all those 3. You must harmonize those 3 in order to have a better harvest. Because sometimes for one disease, we'll have a different management practice or management method than the other. So later on, I will show you the difference between brown spot and bacterial leaf blight. Okay, and this deferred inspection follows a set of rules. It's called Cox postulate. Okay, so this is a systematic method in identification. So why do I say that it is systematic? Okay, how many pathogen groups do we have? We counted how many earlier? Fungi, fungicide, bacteria, phytoplasma, virus, viroids, nematodes, phanerogams. Actually, there is also a prokaryote, right, ma'am? Yeah. But it's very difficult. In the Philippines, there are no one that studies that right now. So there are a lot of pathogen groups. And will you test everything for everything? If you want to know the pathogen or the disease, is there a need to test for everything? For the virus, for the mycoplasma, viroids, etc.? Is it economical or practical to test everything? I will change the question. It's not practical because each of the different tests will require a lot of time, a lot of money. For each of the different tests, there is expenses. Okay, especially if you will be going to the testing facility or the test clinic. Okay, everything costs money. Okay, cost time and cost money. So it's not practical. So you must follow a set of rules so that you will be guided. Okay, if this looks like a fungi, then I will be performing the procedure for fungi. If this is a bacteria or this looks like a bacteria, I will be going for bacterial identification. That way, you will be saving a lot of money, saving a lot of time. Okay, next slide, please. So the Koch's postulate has 4 steps. First is association. Association is when the causal organism is associated or found in the plant or plant part. Okay, so there is association between the causal organism and the host plant. If there is no association, then will you follow the Koch's postulate or will you proceed with your diagnosis? If you cannot find any causal organisms, no, because if you follow the procedures, we will not get anything. Okay? Next is the isolation. So isolation is the separation of the causal organisms and growing into pure culture. So this is 2-part. So you first isolate or separate and then you perform purification or you produce pure culture. That way you will identify or characterize that pathogen. If it is a fungi, you characterize the fungi. You look at the growth, you look at the spores, you look at the size, the color, etc. That way you can characterize that fungi. For bacteria, you also look at the colony characteristics. What is the color, what is the size, what is the form, what is the elevation. So that way you can characterize or you can identify that causal organism. So do you think it's enough to give the management? No. We are called pest clinic because we are plant doctors. Okay? If you go to a doctor and the doctor says that, you have this symptom, you're sick with this one, then do you do what the doctor says without the testing? If the doctor did not test your blood, your urine, when you have high fever, but the doctor did not test your urine, your blood, will you follow the doctor's recommendation? No, because you are not 100% sure that what the doctor said is true. So you must follow the procedure. So are you getting my point? Okay. Next is the inoculation. So why do we inoculate? So this one we don't do on humans. Okay, we only do this on plants. Okay, inoculation is when you get your pure culture from your isolation, you introduce that to the healthy plant. And that plant should have the same symptom as your original plant. What if the symptom is different? When you get your pure culture from your isolation, you introduce that to the healthy. If the symptom is different, maybe your isolated organism is not the right organism. So you must repeat for example, you have a plate here, then look at that. Maybe you picked the wrong colony. So then you will return to the start again. Okay, so this should have the same symptom as this number 1 association. And then this is the confirmatory part, okay, the re-isolation. So when you re-isolate, you should have the same culture as this one. Okay, that way you are sure that that is the pathogen. If you follow these steps, then that's the point wherein you give the management. Okay? In the pest clinic, we don't just identify the pathogen. We also give the recommendation. Is it also true here, ma'am? Okay, so you don't just give the identification, oh, this is bacterial leaf blight. Then, what will the farmer do with your identification? It's bacterial leaf blight. Oh, so what will I do? Okay, so you always give the recommendation. Because we are plant doctors. Okay, next slide. So, I will only discuss isolation and inoculation. Okay, for isolation, this is for pathogens that can be separated from the host plant. So, what are the pathogens that we cannot separate? Viruses. Viruses, you cannot separate that from the host plant. They are obligate parasites. Rust, we cannot also isolate them from the host plant. Okay, so we can only those that we can separate, we call them facultative subprobes. Those are the ones that we can perform isolation. Okay, so different pathogen groups, different techniques. Like I said before, that's why we are doing it systematically, because different techniques for different pathogen groups. So next slide, please. So first is the technique for fungi. This is the tissue planting technique. Okay, so first you have a specimen, and then from that specimen you clean it up, you remove the debris because those are sources of contamination. Okay, as you can see, you will be growing it in a, in a culture plate or in most cases PDA plate. Okay, and that is a rich source of nutrients. So anything that you put there will grow. Okay, so you remove the contaminants to make sure that you will only get the pathogen or the causal organisms. Then you cut from the advancing region. Okay, so we cut in the advancing region. Why? Why do you think we cut from here? Why not in the middle? Then you cut from the advancing region. Yes, louder. Okay, so we cut in the advancing region. It's faster spreading. Yes, because the pathogen is actively growing there. Okay, if you cut it in the middle, this is where the dead tissues is, and those pathogens will need the healthy tissues to grow. So you need the advancing lesion because this is where the actively growing pathogen is located. Next, the arrow is not here. You surface sterilize. So this is further sterilization to make sure that there are no contaminants. Then you incubate them. Okay. Like I said before, deferred inspection is inspection with incubation. So you incubate, okay, and then you characterize this growth. Okay. So if you see it, for example, for Bipolaris maydis, so what's the growth characteristics of colony characteristics of Bipolaris maydis? Who remembers? No one remembers. Okay. So it's gray and the margin is irregular. Okay. So though each of the different pathogens or each of the different genera has their own characteristic growth culture. Okay. So that way you can identify them. And then you purify. Okay. In isolation, we always purify. Next, please. For bacteria, we perform straight plate method. Okay. So we before we clean, we remove the contaminants, and then we perform bacterial oozing. So this is what is bacterial oozing? This is the only sign of the bacteria. Bacterial oozing is like you will see a milky substance going out of the tissue. Okay, so you will see milky substance going out of the tissue. Okay, so if you see that, then there is bacterial cells on your specimen. So once you get the bacterial suspension, you spread this on the plate. So actually, this is just one plate. This is just showing you how we do strict plating. Okay? With strict plate, you dip the wire loop inside, and then you spread it first on one side, then then sterilize this, then strict the other side, and then strict it again, usually 3 or 4 times. So why do we perform strict plate method, in in 0.1 ml of water, there are millions of bacteria. Okay, and if you just plate it at once, then those millions of bacteria will stick together. And it's very difficult to get a pure colony. Okay, for a pure colony for bacteria, it should come from a single bacterial cell. So how does bacteria multiply? How does bacteria multiply? By cell fission, diba? Division. So one mother will create two cells, and it's exponential. Okay. Actually, in 3 days, I forgot to mention the fungi. For fungi, the incubation time is around 4 to 5 days. Okay. But for bacteria, it's only 1 to 2 days. Okay. In 1 to 2 days, you can already see the colonies. Okay. Then, same as the previous one, you purify. Okay. So this is what it looks like. So if this is the first streak, you can see the bacterial cell is already sticking to each other. So it's very difficult to get a pure culture. Okay, next slide, please. So actually, this one is for nematode. Nematodes are obligate parasites, but we can still characterize them during isolation. Okay, how do you characterize nematodes? So actually, this one is for nematodes using their mouth parts and the morphology, right? Okay. What is common between all of the plant pathogenic nematodes? They have what mouth part? Yes, stylet. Okay. All plant pathogenic nematodes have stylet, and then they have different shapes of the stylet. They have different skins, and then for Meloidogyne, for what else? The cis nematodes, the female forms differently. Okay, so we can still characterize them when we isolate. Okay, so for nematodes, we perform the modified Berman funnel method. So we incubate them. Okay, it's either soil or macerated roots or tissues or leaves, sometimes even seeds. We macerate them, and then we submerge, not submerge, we put them in the setup so that the nematodes can go down, and so they will go down here in 1 to 2 days and can be collected underneath. So the principle is behind that is gravity. Okay, so the nematodes are motile, so they will move outside of the tissue or the soil, and then they will be collected at the bottom because they are motile. The once you collect that, you can look at them under the microscope and you characterize them. Okay, next slide, please. Okay, so that the previous slides are for isolation. We now go to inoculation. So inoculation is the transfer of inoculum. So for fungi, it's spores or sclerotial bodies. For bacteria, it's the bacterial cells. For nematodes, the nematode itself. And for viruses, it's the virus particles. Okay, into a susceptible plant. So it's very important to have a susceptible plant. What happens if you use a resistant plant in your inoculation? There will be no disease. Okay, so it's important to use a susceptible plant in inoculation. Okay, so this simulates the natural infection. Okay, so how does these pathogen groups enter the plant? So next slide, please. So for bacteria, there are 2 modes of entry. First is by natural openings. So we have the stomates, the hydathodes, the nectarthodes, and then they can also enter through the wounds. Okay, all plant pathogenic bacteria are motile. So what part do they have for motility? Right, flagella. Okay, they have the flagella and they move towards the opening. Okay. Next slide, please. For fungi, so they can enter through wounds to natural openings, but they can also enter via direct penetration. Okay, they can easily enter the plant without any help from without any external help. Okay. Next slide. For nematodes, actually, it's the same with fungi. They can also enter the plant via direct penetration. They can also enter the plant via natural openings and also through wounds. For viruses, next slide, please. For viruses, it's different. For viruses, they always need help from from external forces. Okay? They don't have the they don't have the flagella like the bacteria. They don't have the infection peg like the nematode. They also don't have the infection peg for fungi, stylet for nematode, sorry. So infection peg for fungi, stylet for nematode, and then flagella for bacteria. So they don't have that. What are the components of a virus? It's only the DNA DNA or RNA and a protein coat. So they have only two components. They cannot enter the plants by themselves. So we let them inside via mechanical inoculation or, next slide, via vectors. Okay? So the vector will feed on an infected plant. Once it feeds on a healthy plant, it will transfer the virus to that healthy plant. Next slide, please. So for fungi, for those that produces spores, we perform spray inoculation. So first we have the pure culture. We dislodge the spores from that culture, and then this is our spore suspension. We filter that so that you can have the purified spore suspension. Next slide. Then that spore suspension, we spray that on a plant. Okay? And then we cover it with plastic. Why do we cover it with plastic? Okay. So for fungi, they need to germinate first. And for germination, what is the necessary environmental requirement? Moisture, just like the seeds. If there are no moisture, will the spores germinate? No. If it germinates, it will only die. So we are covering it with plastic within 24 hours to make sure that the spore will germinate and will enter the plant. Okay? After 24 hours, we need to remove the plastic because it will have an effect on the plant itself. Okay? We did not create any wounds because the fungi can enter the plants directly. Okay, next slide, please. So for bacteria, we perform the clipping method. Okay, just like in fungi, we perform purification of the suspension. So we have the bacterial culture. We put water, we dislodge them, and then we remove the bacterial suspension. And that bacterial suspension, we dip a scissor, a sterilized scissors, okay? We use sterilized scissors to make sure that it is not contaminated. And then we clip the tip of the leaves. Okay, for those that are planning on doing their thesis for bacteria and for rice, then this is the method, okay? Okay, you dip and then you cut. Okay, because how does the bacteria enter the plant? Only by natural openings and wounds. If you create wounds, then they will enter there. Okay, it's very because it's for bacteria, it's more difficult to enter the plant via natural openings because they still need to move, and their movement is very slow. Okay, for bacteria, we don't cover it with plastic. Okay, because we already let the bacteria in via the wounds. Okay, next slide. For nematodes is the easiest part. Okay, So if you want to go to nematology, this is the easiest inoculation. Okay, you just have an infected roots, and then you put it in the soil near the healthy plant. And then in 2 weeks, you will observe the root galling. So this is for root knot nematode, but it's different for other nematodes. Okay, but for soil inhabiting nematodes, then this is the general method. Next is the viruses. Okay. Next, for viruses, this is tobacco. Okay. So what is the disease of tobacco? Okay, tobacco mosaic virus. Okay. So for tobacco mosaic virus, we use mechanical inoculation. Okay. But for others, for example, for tungro, we perform vector transmission. Okay, but this is easier compared to that. This is easier to do in the lab, actually. Okay, if you perform vector transmission, it will take a lot of time. So we are using mechanical inoculation or transmission. So from an infected leaf, you grind that to get what? To get what? To get the sap. Okay, you get the sap from the infected plant because viruses are obligate. It will not live outside of the plant. And that sap is still part of the plant. So you use the sap for inoculation. You add abrasive. So we are in the Philippines, we are using carbon dust or sometimes volcanic ash. So this will create those tiny wounds on the surface of the leaf. So if there is a sap with abrasive, you just lightly spread it on the surface of the leaf, and then the virus will enter within 5 to 10 minutes. So in 5 to 10 minutes, you clean it up, you rinse it with water, because what will happen if you let this one at the surface of the leaf? It will burn. Yes, this is very hot. Okay, so if you leave it there, if you don't wash it, it will burn. So in 1 week or 2 weeks, you will see the infection. Okay, so that is the process of diagnosis. Okay, so from association, isolation, inoculation, and lastly, the isolation. So now I go to, next slide please, major rice diseases in the Philippines. Okay, so like I said before, I've been working in rice for how long? More than 10 years. Okay, so I think I've been working in rice for around 13 years. It's very long. So am I old? Okay, so this is the major rice diseases in the Philippines. I will only be discussing the major rice diseases because if I will discuss all, it will take some time. And actually, I only have around 15 minutes. Okay, so first, next, so we have the leaf blast. Leaf blast is caused by Paricularia oryzae. Okay, so this is leaf blast, but it will occur on all parts of the plant. It will occur on the panicle. It will occur on the node, on the neck, on the collar. Okay, what is the collar? Where will you find the collar? In the junction between the leaf and the sheath. That's the collar. Okay? So it will occur all throughout the life stages of the plant. Okay? But the most distinguishable characteristic of rice blast is its leaf blast. So there is a spindle-shaped lesion with a grayish center and the dark margin. So next slide, please. Next is sheath blight. So just like the name implies, it occurs on the sheath. The sheath will have an ovoid or elliptical lesion with a grayish or whitish center with a dark margin. Okay, sometimes this occurs on the leaf, but only during conducive environments. Next slide, please. So this is brown spot. So this is caused by Bipolaris oryzae. Okay, so I forgot to mention. So can we go back to leaf blast? Next slide, please. Okay, so leaf blast actually occurs on the field that are high in relative humidity, so around 80 or 90 or above, and a slightly lower temperature, and with no standing water in the soil. So usually upland conditions. So that is where leaf blast thrives. So how will you control them? Leaf blast. So if those are the different conditions for their growth and development, so you just actually it's also prominent during high nitrogen fertilization. Okay, so how do you control? You irrigate and then you optimize your nitrogen fertilization. So next slide for sheath blight. This is fairly common when there is flooded condition and closed plant spacing. Because this is caused by Rhizoctonia solanae. Rhizoctonia solanae does not produce any spores, but they travel via irrigation water and also contact. So most likely, in most of the times, leaf-to-leaf contact. So if there is close planting, there is higher spread of the disease. So how do you control? You optimize also fertilizer. You reduce water. Okay. Kanina is earlier. Okay. Earlier I mentioned in leaf blast you irrigate, but for sheath blight you must reduce the irrigation. Okay. Actually the best management for water is the alternate wetting and drying. Okay. You don't flood continuously. You alternate. And you increase the plant spacing. That way, the spread of the disease will be lesser. Next slide is brown spot. For brown spot, it's different from the earlier ones because for brown spot, this thrives in stressed conditions. So what do I mean by stressed? Usually drought condition, lesser and nitrogen fertilization. So that is the times where brown spot occurs in the field. Okay, so typically upland condition. So how do you control? You irrigate and then you increase the soil fertility. So you put more fertilizer. Next slide. This is sheath rot. So sheath rot you might mistake for sheath blight. But the difference between sheath rot and sheath blight, for sheath rot, in most cases the panicle will not go out of the sheath. Okay, it will not go out. If it will go out, there will be no grains inside. There are no seeds inside the grains. Also, the color. For sheath blight, the lesion is grayish center. But for sheath rot, it's brown on the inside and the margin. Okay. Actually, this also thrives in the same condition as sheath blight. So the management strategy is also the same. Next slide. This is, the earlier ones actually is fungal pathogens or fungal diseases. This one is bacterial pathogens. In the Philippines, actually, there are two bacterial pathogens that are prominent. Two? Yes, two. Okay. They are from the same species, but they have the different pathovar. Okay, so this one is caused by Xanthomonas oryzae, pathovar oryzae. Okay, so you're still remembering your crop rots. Yeah. Okay, so leaf blight, it occurs most of the times from the edge. So if it occurs on the edge, where does it enter the plant? Is it the stomates, the hydathodes? Is it the stomates or hydathodes? What is present at the edge? Is it the stomate or hydathode? Hydathode. Okay, so that is how Xanthomonas oryzae, pathovar oryzae enters the plant, usually via the hydathodes or through the wounds. That's why sometimes you can see them in the center. So this actually spreads during high nitrogen fertilizer. There is also flooding conditions, and there is an increased spread when there is typhoons. Because what happens during typhoons? What happens to the leaf? The leaf are swayed very violently, and there will be contact, and there will be wounding. Okay, so if there is wounding, there is contact, then the pathogen can transfer to the other path, to the other plant. Okay, so just like shift blind, the management strategy is lesser optimized fertilizer, and then lesser irrigation. So next slide. So actually, I did not mention so let's go back first. Okay. So the other one, so what is the other Xanthomonas oryzae? What is the other path of Xanthomonas oryzae? Oryzae? Oryzicola. Okay. So it's Xanthomonas oryzae pathovar oryzicola. Okay. So the symptom or the disease is bacterial leaf streak. Okay. Streak because you will see it inside the lamina. Okay. There will be streaks. So if it's in the lamina, how does it enter the plant? It enters via the stomates. Okay. Leaf blight via the hydathodes. Okay. Bacterial leaf streak via the stomates. Next slide. This is my last slide actually. So the most prominent viral disease in the Philippines is tungro. And I think even here, it's also tungro. Okay. So tungro is caused by what disease? What pathogen? Rice tungro. I think even here, it's also tungro. Okay, I heard the end. Okay, it's very important to hear the end. It's caused by rice tungro bacilliform virus and rice tungro spherical virus. Okay, so there will be no symptom if it is not caused by those two. For spherical, it can be transferred all on its own, but for rice tungro bacilliform virus, it cannot be transferred by its own. It needs the RTSV to be transferred. Okay, but if it's only RTSV, there will be no symptom. Okay, are there any cure for tungro? Are there any chemicals to control tungro? No. You control what? You control the vector. Okay? If you control the vector, you will control the spread of the pathogen. But how will you control the pathogen, for example? You just do roguing. Okay? You remove the infected plant and you destroy them. Okay? In some farmers, they just pull it up and then it leaves it at the side of the field. Is it okay? No, because it's still a source of infection. So you should at least burn it or you put it under the ground. Okay? So that's all. Thank you very much. Is it okay? No, because it's still a source of infection. So you should at least burn it or you put it under the ground.
Nggak kerasa ya, lama juga ya, tapi semoga masih tetap semangat. Okay, question please. We still have how many? 5. Okay, question. Kerasa ya, lama juga ya. One. Okay. Semoga masih tetap semangat. Okay, first. Question please. We still have how many? 5.
I want to ask you, there is typhoon in Philippines. Is there typhoon in Philippines cause spreading the pest and the disease is more spread and rapid, more wide and rapid? And how do our friends in Philippines to overcome the damage after the typhoon?
Actually, also typhoons in the Philippines. Okay, she is asking if there are typhoons in the Philippines that can spread the pathogen or the pest, and are there any measures? Actually, there's also typhoons in the Philippines. But the problem with typhoons, you cannot control them. That's why it's a natural phenomenon, a disaster. Okay, so if the typhoon hits, then there's no cure for that. So usually when typhoon hits during the susceptible stage of the plant, then we don't have any control measures, or we don't have any ways to counter that problem.
Or just escape when
Yeah,
you know that
escape.
this month the typhoon will come and then just remove something or how?
Yes, that's actually one good tactic. If you know the months that the typhoon will enter the area, so that's called disease escape. You just adjust your planting schedule. For example, if typhoon season hits around June or July, June or July, and your susceptible stage is around 30 days after transplanting. So you transplant, for example, earlier. That way, your crop will not be susceptible. Okay, because in diseases, actually, there is adult resistance. This is especially true when it comes to toro. Okay, adult resistance. If you already escape or the stage that is susceptible for that plant, then there will be no or lesser disease. Especially true when it comes to toro.
The next question, spreading the pathogen. Is it possible typhoon will spread the pathogen?
Yes, ma'am. Especially when it comes to airborne and soilborne and waterborne pathogens. Okay, because in typhoons, there's a lot of rains, there's a lot of wind, there is faster spread of the pathogens, airborne or waterborne. So for bacteria and for rhizoctonia, they are both waterborne, so they can spread easier during the typhoon. Actually, it's different when it comes to the insects. When it comes to typhoons, when there is typhoon, insects will disappear. But for diseases, when there is typhoon, there is faster spread.
Spreading. Okay, thank you.
Actually, it's different when it comes to the
Next,
insects.
please.
When it comes to typhoons, when there is typhoon, insects
One.
will disappear. But for diseases, when there is typhoon, there is faster spread.
Next, please.
So, mungkin aku menjelaskan di bahasa Indonesia dulu ya, Bu ya. Nah, sebelumnya saya waktu itu peraja di Balai Penyuluhan. Nah, biasanya kalau petani itu dikasih resep pestisida gitu ya, Bu. Jadi petani tinggal mengaplikasikan aja gitu. Petani yang awam seperti itu. Kalau petani yang udah jago itu beda lagi. Nah, as you mentioned before, sir, that each symptoms, pathogen and pest has different kind of handling and management, and it must be really difficult for new farmers or maybe each symptom, how is the mechanism that provided by governor management, or maybe even there are a private institution that can help civilian farmers or newbie to solve any kind of disease or even there are a private outbreak spread in the country, or even maybe any clinics that refers to this lesson.
Okay. So yes. So I think you already had IPM courses, right? Okay. So in integrated pest management, actually that's also part of the tactics when it comes to identification. So in the Philippines, actually there is one special program that they do so that the farmers can be more knowledgeable on these different pests and diseases, not just rice, even other crops. It's called the Farmer Field School. It's also present here in Indonesia. Okay, so Farmer Field School. What happens in the Farmer Field School? those experts, so they can be experts, they can be technicians coming from the governor, from the municipality. Those can train the farmers that are members of that farmer field school in the identification. So in identification, not just for insects, but also for diseases. In the Philippines, also weeds. They also consider weeds during that identification. The sad part is in the Philippines, most of those that can go to the farmers are actually from the private companies. So private companies like this in Indonesia, I think it's very, very common. So in Indonesia, I think even the technicians from the companies, they will go to the farmers, and they are because they are promoting their products, and they are also able to identify these different pests and diseases. But the problem with that one is because they are bringing their chemicals, then that is what they recommend. So the best method is if there is a bigger push in the government side or in the local government side for their own technicians to be able to establish this small farmer field school, because farmer field schools are actually very good in making farmers more knowledgeable on these pests and diseases. Okay, actually, farmers are, but we as the expert don't claim that we know all of the things in the farm, because in the farm, farmers are still more knowledgeable. We might know this one, the identity. We know this is the management strategy, but because farmers are facing these kinds of problems day by day, sometimes they have their own knowledge, or what do we call it? The indigenous knowledge. Okay, even their management strategies, you can find they do some different things to control this pathogen. In most cases, it's the vertebrate pest. They have a lot of indigenous knowledge for those different types of pests. So the best method still is the collaboration. Okay, you have the idea, they have the indigenous knowledge. You must combine them so that there is more harmonious management. Yes. Yeah, I think it's the same in all Southeast Asian countries.
Okay. Jadi ada seperti kita ya, yang Farmer Field School, PHT, sekolah lapangan PHT. Sekolah lapangan PHT, ada petani di situ yang belajar, dan selalu tadi disebutkan farmers is the expert of their field. Itu yang ada important point. Jadi harus ada kolaborasi antara farmers and expert yang lain untuk mengkombinasikan the indigenous knowledge, pengetahuan apa istilahnya Pak Herman ya? Indigenous knowledge itu kearifan lokal yang mereka sudah punya, dikombinasikan dengan pengetahuan yang kita punya. Itu akan meningkatkan bagaimana mengelola pertanian mereka. Thank you so much for that. Oke. Kira-kira begitu ya. Nggak apa-apa. One more question. Mengelola pertanian mereka. Oke. Yang di depan. Oke. Kira-kira begitu ya. Nggak apa-apa. One more question. Oke.
Thank you for the lecture. I would like to ask a question. Are there any rice diseases or pests that the Philippine government is currently trying to prevent its entrance to the Philippines border? Thank you. Thank you for
To the entry to the what?
Are there any rice diseases that
The quarantine. Okay,
that
quarantine.
isn't present on the Philippines borders.
Okay, actually, for the Southeast Asian countries and East Asian countries, we only have one quarantine pest for rice. It's called Udbata disease. It's caused by Aphelis oryzae. Okay, so I think that's the only disease that we don't have in the Philippines currently for the field crops. But for seed crops, it's for seed rice, it's different. Do you have Aphelis, ma'am, here? No, no. I think no, because Aphelis oryzae is, I think, is in Cambodia and in around East Asia. Yes. Do you have Aphelis, ma'am, here? No. Fortunately, I was in before. I thought I was in Erie, so I'm more familiar. Yeah, it's called Udbata disease. If you can look it up. So, what happens in Udbata disease is the seeds looks like cattail. Do you know cattail, the plant? There's no. Usually, I draw in a board. Okay. So it becomes cattail. Instead of the panicle spreading, it will just be lumped together, and then there will be no seeds inside. So it's still not present in the Philippines, and I think it's not present in Indonesia. Because in my previous work, I was checking where these different diseases are located, and I think Indonesia is not part of where that Aphelis oryzae is located. Because in my previous work, I was checking where these different diseases are located, and I think Indonesia is not
Until
part
3
of
o'clock
where
also.
that Aphelis oryzae
But now we still stay here until 4 o'clock. Actually, we planned only until 3 o'clock. But thank you so much for your attention. And we have a small souvenir. Okay.
Okay, I would like to say thank to everyone here. Terima kasih sudah hadir di acara dosen tamu. Ya, semoga apa yang diberikan oleh para dosen dari UPLB ini bisa menjadi landasan untuk take off dalam melaksanakan mata kuliah klinik tanaman. Yes. But first of all, is it possible for my student here to contact you by email or so? Is there any email address if someone want to maybe student exchange? Yes, of course, Ibu Evi. Who wants student exchange to UPLB? We can start because the MOU will be renew MOU between IPB and UPLB. So if there. Because in this one is no email address of you. If it is possible, then I will. Yes.
Yes, actually, yeah, we don't have an email address here, but we actually have an email address in here. You can just address us. So the official email address of our institute is iweb.uplb@up.edu.ph. Okay, if you email that and then you specifically mention us, then they will forward your email to us.
Okay,
then when they forward their email to us, then we can contact you after.
Okay, thank you. Because sometimes here in pump protection, the operator will be need more encouraged to send or forward the email which is come, and then that's the difficulties. But please, kalau siapa yang tertarik, if you want to, you can ask me or Ibu Evi to make a further collaboration or further discussion with all of them. Yes? Okay?
Yes.
Thank you, Bethany.
Okay. And we have a small souvenir, traditional weaving from Sumba. Just
Everyone here, there is another doctor here who work or interest in wheat scientist. Actually, Dr. Nina is a wheat scientist, and because the wheat is not in here, So Dr. Nina already talked with other expert in agronomy, yes, agronomy and horticulture. But if you want to know about weeds more, then contact Dr. Nina. Okay. Dr. Nina already talked with other expert in agronomy, yes, agronomy and horticulture. But if you want to know about weeds more, then contact Dr. Nina. Okay. Dear, there is a souvenir from Professor Hermanu. So he promised, and then I will give to you. Sorry, he is not here. Dear, there is a souvenir from Professor Hermanu. So he promised, and then I will give to you. Sorry, he is not here. This is a special from other province, right? So everyone very, just put it, everyone here very, because this is special. That's good, yes. That's what the color. This is a special from other. Cuma gini aja kan? So everyone very, come. Just put it, everyone here very, because this is special. That's good, yes. The color. So the doctors here invite who give a question, so to come up and come here to the in front of. Siapa yang bertanya? Tadi yang bertanya boleh ke depan untuk difoto bersama. Silakan ditunggu. Siapa namanya? Siapa yang bertanya? Gak ada yang mau ke depan. Tadi yang bertanya siapa? Untuk ke depan silakan. Yuk, biar cepat yuk. Siapa tadi yang bertanya? Gak ada yang mau ke depan tadi yang sudah bertanya ya kemana? Nah, terima kasih. Oh, kamu Christian nanya? Oke. Faiz nggak nanya? Faiz tidak bertanya. Yang sudah bertanya ya kemana? Faiz tidak bertanya. Terima kasih Faiz, karena kemarin Faiz nganter-nganter Dr. Nina pergi ke agronomi dan hortikultura. Yes, Ibu Nina, Dr. Nina, because Faiz, one of the student help you to. Dr. Nina pergi ke agronomi dan hortikultura. Oh, why don't you say that?
You can also, if you graduate and you want to go to masters, you can go there, and Dr. Nina can handle masters students. So you can go for with science. Also, if you graduate and you want to go to masters, you can go there, and Dr. Nina can handle masters students. So you can go for with science.
terima kasih. Tidak apa-apa kalau sekarang mau istirahat, kemudian jangan lupa sholat asar, karena kemudian akan dilanjutkan dengan acara pertemuan dengan beberapa teman yang sudah diminta untuk berkumpul dengan ini, untuk berdiskusi tentang teaching modalities dan lain-lain. Jadi saya cukupkan untuk acara dosen tamu pada mata kuliah klinik tanaman. Namun ada pengumuman, saya membuka 4 mahasiswa yang akan yang kalau berkenan volunteer, karena mereka besok mau around the Bogor, jadi perlu guide. Siapa yang mau ya, jadi siapa saja nanti 4 orang silakan menghubungi saya ya. Ya, terima kasih. Jangan lupa isi presensi. Jangan lupa isi presensi. Assalamualaikum warahmatullahi wabarakatuh. Terima kasih kepada Ibu Evi. Terima kasih Dimas. Terima kasih Farhan sudah membantu. Sampai jumpa hari Jumat pagi minggu depan untuk mulai praktikum dan responsi. Bye.